Hi, I'm Sharon Gusky, Professor of Biology at Northwestern Connecticut Community College, and I'm here with Kelsey Sudol, Adjunct Professor of Biology, also at Northwestern. We're going to be talking today about using virtual citizen science activities to introduce students to careers as research technicians. This is part of our NSF-funded ATE project, engaging students from classrooms and camps to college and advanced technical careers. Next slide.
We use two main platforms in order to do this, Zooniverse and SciStarter, which you can, links to them are on the bottom of the screen, and we did multiple projects on each. And we're going to highlight three main projects today. First is Etching Cells, which is through Zooniverse, and then Water Insights, which is actually a citizen science water quality project that was developed by a student in India, a high school student. And then the last project we'll highlight is Star Catchers, which is through SciStarter.
So the project I'm going to talk about is called Etching a Cell. So before doing this project, students learn about the various types of microscopes, including electron microscopes, and they learn about the different parts of a cell. So what this project does is it introduces students to what a research technician will do in a lab, and the students take a look at mitochondria inside a cell by looking for mitochondria in these electron micrographs. Students also learn about the researchers involved. And they also start to identify themselves as scientists when they see that the researchers, like this here, this is Anne, and she's actually segmenting it, an image in the lab, do basically what they're going to be doing, but they're going to be doing it at home on their computers. So here's a look at the project itself. So students come in, they read the field guide, which is here on the side. It takes them through the mitochondria gallery, so they have an idea of what the different mitochondria look like. It takes them through, if there's no mitochondria, what those images look like, gives them information on common challenges, and shows them how a nice drawing or etching of the mitochondria looks like. They then click on the pen. They have these images, and they start to identify mitochondria and etch around them, and my etching is not very good. But the students, it takes a little practice to etch. They etch around, they do a count, and then when they're done, they click done. And then they start to contribute that data. So they would etch all the mitochondria that they see in the field, and then they would get another field to do. So this is their contribution to this large research project. We also have them work and collect their data and share their data with each other and do a discussion about their data. They answer some specific questions that are specific to our course and the information that we want them to learn and understand. And then what's really exciting for the students for this project is that once the project is finished, you can see that this project has already been finished. They get their name listed as a contributor. So they actually get their name published on the website as a contributor, and they can mention this when they talk about what they've done in their science classes. They can give them the experience of being a research technician and collecting and analyzing data. So next, Kelsey's going to talk about Water Insights.
The next project we're going to talk about is Water Insights. This is a water quality national database created by a student in India. She's a high school student who saw that her local pond was catching on fire, and she wanted the ability for her community to take data in order to better understand what was happening. And she then created this platform for more students across the country to do. What was great about this project is it was part of actually sending some projects home to each of the students' houses for them to do. So they received what you see here on the left, which was a small kit where they had 2 water testing strips, and they were allowed to choose whatever type of water they wanted to do. Most students, as you can see through their the data here on the right, did choose their tap water, but we did have some students decide to do snow melt, and a few did some stormwater runoff or their local pond, but it's not listed here on the right. So what was great about this is the students were able to do a simple hands-on activity. They take their water, they dip the testing strip here on the left in that water for about a second, and then they leave it to dry for around 30 seconds, and then they have to check their data against the chart that we gave them. And they got to test 6 different parameters of their water. Before they actually did this lab, they did a pre-lab assignment where they learned more about specifically pH, which was one of the parameters that they were testing, as well as some background information on the project and just on general water quality. After they did this initial lab and they submitted their data, we took all of the data for the entire class. It was actually 2 sections of the class, and we gave it back to the students. And we asked them to talk amongst themselves through discussions in their lab teams. And they also talked to the whole class, and they got to compare their data. And they also got to see the differences between those data. And they had to do a final report in which they analyzed their data at the class level, so comparing it to the class, at the national level, because you can submit your data to Water Insights as a whole and can look at everyone's data from across the country. So they had to compare out-of-state water quality data. And then they had to come up with some possibilities on why their water might be different, what can cause some differences between their water. And I had several students say specifically they'd never even thought about water came out of their tap. They were completely surprised that their water was not exactly a pH of 7. They had no idea that they could be drinking slightly acidic water, and it was really surprising for them. But they actually appreciated that kind of insight on their own home water and learning about how everyone's water is different and that it takes a lot of community effort to monitor and protect our water quality. And they were excited at the thought that their data could help create a database for Connecticut specifically, because we are the first, we had the first data point submitted for the state. Uh, and so they then had to do a post-lab quiz afterwards, but the report kind of tied that all together. But the next project we're going to talk about is stalk catchers. So for stalk catchers, which is the next project we're going to talk about, we thought we would show you an example of the weekly folder for each activity. So in this folder, you can see that we would put a weekly video where we go through all the different material that they have to do for that week. And they have a pre-lab assignment and discussion where specifically for stalk catchers, they would discuss and learn more about osmosis and diffusion as well as capillaries in order to better understand the lab, which I'll talk about in a second. They have the specific lab folder assignment and then a post-lab folder as well. So students would go to the crowdsourcing platform Stall Catchers, and here they would identify stalls in mice brains. So they have small videos like this here on the left, where they either have to identify whether the blood is flowing completely through a highlighted area here, or whether it is not flowing. And they have a short video where they can see the blood flowing, and they just have to click whether it's flowing or stalled. And often underneath, there's also advice from other people who have looked at it, and they can compare their answers. They then have to track for 20 of these videos whether they saw a stall or not, and then they have to calculate whether their total percentage of stalls. After they completed that lab, their post lab was actually to put all of their data together with their lab groups, and then they did instead of a lab report, they did a research article in which in 1 to 2 pages, they had to write a news article on background information about stalk catchers and Alzheimer's. And they were able to think through the lens of giving a the normal general public this scientific information. And I know for several students, it was interesting for them to think about that perspective and think about trying to give what they've learned back to the general public. So after they completed that news article, students had the option for their midterm to actually do a full poster for one of the projects that we've talked about. So for their midterm, students then got to choose a project that we had done by that point to do a poster on with their lab team. And some of the students chose stall catchers. So you can see an example of a poster on screen now. And what was great was that this gave the students an opportunity to go back and review a previous project they work on, but also, again, look at how do they present scientific information to the public. They got to learn how to write an abstract and also think about what images would pop and make people interested in looking at their research, as well as learn how to do a well-designed graph for the general public to read. Uh, this is another example of one. And again, learning how to address the general public while also being scientifically accurate, where to put your hypothesis, your abstract, etc., and have the correct flow of information. What was great is that this again gave the students a chance to collaborate, and they'd sometimes discuss how certain projects were impactful to them. I know several students talked about how stall catchers meant a lot to them, that they were able to contribute to that scientific body of research because they had members of their family that had Alzheimer's, for example, or other students who had well problems in their house were really excited to contribute to Water Insights as well. And to be able to see that interplay amongst the lab teams as they did these assignments was great to see.
So one of the reasons that we picked citizen science projects for our students to do during the pandemic was that we wanted them to have an authentic research experience and introduce them to careers as technicians. Before the start of the course, we surveyed 100 students, this was in our 3 biology sections, and asked them if they're familiar with any of the following technical careers. And we had environmental technician, biotech, chemtech, engineering tech, research technician, and sterile processing technician. 72% of our students reported that they were not familiar with any of the technical careers, and only 28% of the students reported familiarity. And you can see each of those got fairly low scores. So what we did was that we sent our students as one of their extra credit assignments to go and explore technical careers. And I know most people think that extra credit, not a lot of students do it, but we found that extra credit is something that all the students do. And even a higher percentage complete those in some of our assigned assignments. So this worked well. So we're going to show you that site that we went to and talk about what we had our students do.
So we would send students to the Careers in Biotechnology page that you see on screen right now, and they could scroll down and choose 2 technician positions that they were not familiar with, and they got to click and learn more about them and learn also how much would you make in this career? What level of education did you need to do this career? I know several students made comments that they didn't realize they could go into fields like this with an associate's or even bachelor's. They thought you'd have to go higher in education. But the students would have to do an initial post with that information for 2 careers, and then they got to comment on each other's posts about the different information and careers that their classmates chose. And what you see on screen right now are some of the comments that the students made on each other's posts, um, of things that they were interested in or were new information to them. So several of the students talked about how a lot of these positions they'd never heard of are actually really integral to society, specifically with food technician jobs, health and safety technician jobs, and they'd never really thought of them before. And a couple of them commented how a lot of these positions were actually essential workers, and that in the last year, especially with a lot of restaurants shutting down and a lot of food chain problems, that they were much more important than they'd previously realized. And one student actually commented how the water quality technician position, she related it to the Water Insights lab that we had done in previous weeks, and specifically commented how she'd appreciated that job much more after doing the lab because she understood the kind of work and thought and analysis you have to do. And then another student commented how a lot of these jobs, again, seem very essential, and she'd never thought of how much that these people would make, and that the salary aspect was something that was interesting to consider with the amount of schooling you have to do with them. But so it was a really engaging activity where the students got to have different discussions on these different technician positions and learn more about different jobs that they can do in the future.
Well, thank you so much for attending our presentation. Please reach out to us if you have any questions or would like some more information.